Okay, hi everybody, and welcome to this week's Closer Look Journal Club in Biomechanics. This, I believe, is our 9th installment in the fall sort of seminar series. My name is Matt Bursey. I'm at Washington University in St. Louis, and I'm joined by the co-hosts for this journal club, Manuel Rausch at UT Austin, Emma Lejeune at Boston University, Johannes Weikemeyer at Stevens Institute of Technology, and Adrian Braganza-Tepla at Purdue University. So welcome. For those of you who are on the call and for those of you who are watching on YouTube. The purpose of this journal club really is to bring together researchers in the biomechanics community to talk about some of their sort of recent work that's been published in the literature and give us as the audience the ability to speak and engage with those speakers sort of in detail about their paper. And so today we have the pleasure of hosting Professor Bart Spronck from Maastricht University in the Netherlands. Bart received his bachelor's and master's degree from the Technical University of Eindhoven before doing a small stint as a visiting scholar at Macquarie University in Sydney, Australia, sort of a globetrotter of science. After that, he actually received an individual prestigious Marie Curie Fellowship. So he was able to self-fund a postdoctoral fellowship at Yale University in the lab of Jay Humphrey, where I met Bart. And then since then, he has now returned back to Maastricht to start his own research group in the Department of Biomedical Engineering. Bart's work is largely focused on arterial stiffness and hypertension in humans and in small animals. And so today he's going to be presenting about some of his recent work entitled Excessive Adventitial Stress Drives Inflammation-Mediated Fibrosis and Hypertensive Aortic Remodeling in Mice. So with that, I will yield the floor and let you take over, Bart.
Thanks, Matt, for the great introduction. So yeah, I'm Bart. I'm in Maastricht currently. It's 5 p.m., so it's getting dark here. So indeed, adventitial stress, fibrosis, and hypertensive aortic remodeling. So just to clarify what I mean with adventitial stress before we lose people down the road. So we're talking about the aorta here. The aorta is a large vessel. I'll be looking at mouse aortas here. And the wall of the aorta has 2 main layers, namely the media and the adventitia. So we're talking about the adventitial stress here, and fibrosis is a process whereby collagen forms in an unnatural way, typically due to inflammation in this case the adventitia. Hypertension is of course high blood pressure. And the aorta, we all know, is the largest vessel of the human or mouse body. So this is the paper that I'm supposed to talk about today, but I actually want to go one step back and tell you what led to this paper. And I want to take you along a quite nonlinear trajectory that led here. So these are 2 papers from people who may or may not be on the call. The left one from Jacopo Fruzzi, the right one from Matt. And so the left paper talks about actually about a mouse model of aging, about aging of the aorta. So fibrillin 5 is an elastin-associated glycoprotein, and elastin is the main elastic component of the artery wall. And in human aging, because of the many cycles, and because of the pulsatility, this functionality is lost. And elastin gets fragmented. However, mice typically get way less old than humans. And therefore, the problem is if we want to study an aging phenotype in mice, we cannot do that by simply chronologically aging them. So we have to do something to them. And a good model is knocking out fibrillin 5. So knocking out fibrillin 5 sort of leads to a human mechanical aging phenotype in the aorta. As you can see on the top there, the pressure diameter relationship gets steeper, so we have a larger structural stiffness in these knockout mice. And that's what Jacopo presented in this paper. So on the right, we have a paper about hypertension. And in this paper, Matt and others at Jay Humphrey's lab subjected mice to an infusion of angiotensin II. So angiotensin II is a peptide that causes peripheral vasoconstriction and thereby hypertension, but also causes aortic remodeling and fibrosis, particularly adventitial. So on the left here, you have a mouse aorta before infusion, and on the right, you have an aorta after infusion. And what you can appreciate is that is the very thick outer layer, very thick adventitia in these vessels. And so when I was at Jay's lab, actually what Jay proposed was, hey, let's combine these two and look at hypertension together with aging. Sorry.
Can I ask just one quick question? So when you have these 2 bullet points here, is the fibrosis due to angiotensin or due to the hypertension?
That's a very good question. It is due to both, I would say. But we can talk about this for a very long time. It's a very good question. But I've seen that hypertension per se is not sufficient, and also that angiotensin 2 per se is not sufficient if you reduce the blood pressure. So yeah, both.
Okay.
Good answer.
Okay.
But
cool.
that,
thank you.
Yeah, good question. So basically, and when I was with Jay Humphrey in 2017, he said, okay, let's combine the two. Let's study hypertension in aging. I said, okay, great, sounds good. So what we did is we took those aged mice, fibulin 5, or aged, I should say, so fibulin 5 knockout mice, and subjected them to an angiotensin infusion. And basically what we got was this. So these are the wild type mice. These are not the knockout mice. This is just control mice that we subjected to angiotensin infusion. And I want you to compare the untreated and the Ang2 treated mice here. And what you see is actually not much. We don't see the expected fibrotic remodeling that Matt showed. So as a reminder, this is what that study showed. So a much bigger change in adventitial thickness. So the question is, what's going on here? Why was this not showing the expected outcome? Was the age different between the mice? No, it was not. Was the sex different? No, they were both males. Was the angiotensin 2 that we used perhaps different? No. Was the same supplier, same way of administration. We checked whether it was frozen versus freshly mixed. We checked buffer solution. Was it perhaps Matt who stressed out the mice more than I did? Well, could be. I don't think so. Anyway, many options that we ruled out, and we couldn't really find anything until we thought about, okay, what is the origin of these mice? So the mice in Matt's study were actually directly bought from Jackson Laboratories. Whereas the mice from our study were bred in-house at Yale, and they were bred from mice on what's called a mixed background. So they are not from a pure inbred mouse strain. They're actually a combination of 2 mouse strains. In this case, C57 Black 6 and 129 S6. So we decided, okay, if all of the other things do not explain the difference, then maybe this explains the difference. So we decided, okay, let's study those 2 pure strains, namely C57 mice and 129 S6 mice. And does that explain the response? And if so, why is that response different between those groups? So that's actually the lead up to this study that I'm presenting now about the excessive adventitial stress driving inflammation-mediated fibrosis in hypertensive aortic remodeling in mice. So what did we do? We took mice of these two strains, as I just explained, and subjected them to 2 treatments, namely no treatment and 14 days of infusion using angiotensin 2. Then we euthanized the mice. Before we do that, we measure their blood pressure, by the way. Euthanize them, take out their descending thoracic aorta, and perform what we call quantitative histology. So look at the structure of the wall and the constituents in the wall, but also perform passive and active biaxial biomechanical testing. After that, perform bulk RNA sequencing for the entire DTA tissue. So I'll talk a bit more about the passive and active biaxial biomechanical testing. Some of you may be familiar with it, especially the people who were at Jay Humphrey's lab. but just for completeness. So what we do is we take out the aorta from the mouse, we clean off the excess tissue, we suture all the side branches, and we mount it between small glass pipettes. And what we can do then is you can inflate it and we can actually extend it. And while we do that, we can also measure the diameter and measure the longitudinal or axial force. So what we do then is we run through a 7-step protocol. So we first inflate the vessel at 3 constant levels of axial stretch, which gives us, for example, this pressure diameter, this inflation curve. And then we turn things around. So we keep pressure constant for 4 levels and gradually extend and retract the vessel in the other direction. So we have really a biaxial testing protocol, which gives you, for example, this force stretch curve. So we have data from 7 protocols, and what we then do is we fit that to a strain energy function, which has 2 parts, namely an isotropic part on the left and a fibrous or anisotropic part on the right. And the main thing of importance here is that people have shown that this W, this function, captures and describes arterial mechanics well. So we can use it as a integrator for all of these individual protocol steps, basically. And because it describes well, we can actually use it to take derivatives, so we can get stress and we can get material stiffness from this strain energy function. So conceptually, on the top right, we see the circumferential stress stretch plot. So the tangent of this plot is conceptually the material stiffness in circumferential direction, whereas, on the bottom right plot, the tangent is the material stiffness in the longitudinal or axial direction. So the other thing we do is what we call an active protocol. So we also see whether the vessels can actually contract. So we keep the vessel in this device at a fixed pressure of 90 millimeters of mercury and subject it to different stimulations. So the first one is potassium chloride, which causes membrane depolarization. So you see the contraction, you wash it out, the gray bar. We then subject it to angiotensin 2, the same angiotensin 2 that we indeed use to infuse the mice. And you can see that the aorta shows a transient contraction to that. Wash that out again, and then contract the vessel by phenylephrine, study endothelium-derived vasodilatation, and finally block that dilatation using L-name to get the vessel to basically a maximally contracted state. So this is what the results are looking like. So these are the control groups for the C57 mice on the left and the 129 S6 mice on the right, using 3 different stains. One is called the Verhoef van Gieson or VVG stain, which stains the elastin fibers in black. And we have a MOVAD stain, which we, among others, use to look at the cytoplasm content in purplish here, and to glycosaminoglycans, but you cannot see them on this slide here. And Picoseries red, which stains the collagen, and which we look at using polarization microscopy. So in the control vessels between the 2 mouse strains, there's not a lot going on. However, after angiotensin infusion, indeed, we see in the C57 mice the marked thickening that Matt also observed. And in the 129 mice, we don't see any thickening. And the thickening is indeed mainly collagen, as you can see in the bottom. So we can further quantify this by using colorimetric analysis on many of these images that you see on the left. And indeed, you see, of course, the C57 aorta showing the biggest thickening, and also above the dashed line, you can see here the almost entirely collagenous adventitia of the C57 Ang2 infused aorta. Well, then we also looked at, okay, what type of collagen fibers are deposited. So using this polarization microscopy, by looking at the colors, you can get some idea of whether the fibers are actually thin or thick. And it's mostly thick fibers that were deposited in the adventitia, as you can see here. And this is all despite is the same increase in blood pressure. So both of these mouse strains in terms of blood pressure, this partial answer to your question, Adrian, previously. So the blood pressure was not the differentiating factor between the mouse strains here. They were both similarly increased. So then we looked at the inflammation in the wall. So we stained for CD45, which is a general inflammatory marker. And again, we see that indeed there is marked inflammation in the C57 adventitia, but not in the 129 adventitia. And you can also quantify this and find it back. But where it gets really interesting is where you correlate the inflammation with the wall thickness. And then indeed you find a correlation, which we interpret as the more inflammation there is, the more exuberant the wall thickening is. So indeed, there's a correlation between the inflammatory response and the thickening response. So then we move on to the biomechanical data. Well, there's of course 10,000 ways to plot this. I'm not going to go through all these plots because we can spend a whole meeting discussing only these plots, but I'll just show the most important results here, which are that there's not much going on in terms of diameter, as you can see on the left. But if you look at thickness, again, you see in the dashed line here with the triangle on top, you see that the C57 infused, Ang2 infused C57 aorta is markedly thickened with respect to the rest, and this does not hold for the 129 aorta. And in accordance with that, if we look at the circumferential material stiffness, we see a decreased stiffness materially in the C57 Ang2 infused aorta as compared to the other three groups, basically. Now, this is where it gets really interesting. So if we look at the contraction response, we see actually quite big differences between the groups. So I want you to focus here on the angiotensin II response. So in black, we have the C57 black 6 mice. In gray, we have the 129 S6 mice. So these are the two mouse strains. In solid, we have the control groups, and in the dashed, we have the hypertensive angiotensin II infused groups. And as you can see, there's a big difference, between the two control groups. So the 129s contract much stronger to begin with as compared to the C57s, but also the response after hypertension is way stronger in the 129 aorta. And we can also study this on a mouse specific or individual basis, if you will. So this is going to be a really busy slide, so bear with me, and please interrupt me if I go too fast. So here on this line, we will be looking at only the C57 mice. So the systolic blood pressure was 130 millimeters of mercury, and each blob here is a mouse. So we had 6 mice here. And the gradient, the color in the blob, denotes the amount of contraction going from, as you can see on the bottom here, 100% in black to 0% in light gray. So as you can see, going from no contraction to maximum contraction, obviously the diameter inner diameter reduces, which makes sense because there's a contraction. We can also look at the thickness, the longitudinal stretch, the circumferential stress, the circumferential material stiffness, and the change in stored strain energy over the cardiac cycle. And what's going to be really interesting here is this circumferential stress plot here. So we'll look at that also in comparison to our next group, which is the angiotensin II infused groups. So again, this is both C57 groups. On the top, we have the group without hypertension, and the bottom, we have a group with hypertension. So obviously, the blood pressure is higher in the hypertensives than in the normotensives. But if we look at the circumferential stress, in the dashed box, what I've indicated is basically the range that the controls were in. And as you can see, after the infusion, most of these mice were outside of the control range, which we interpret as these mice are not able to maintain homeostasis. They are not able to maintain their circumferential wall stress, even given their contractile ability. They cannot adjust to this. They cannot adjust their wall stress back to their control level. However, if you look at the same plots for the 129 mice, so the other mouse strain, we again see one block per mice, and you can see already here the very large contractility that some of these aortas have. Some of them contract back really to almost an inner diameter of 0, so you almost include the lumen. And if here we look at the circumferential stress, we indeed see that most of these aortas are in fact able to control their circumferential wall stress to within the control limits, to maintain wall stress to normotensive levels, basically. And again, so this is a plot that you've already seen of inflammation versus wall thickness. We can make a similar one of contraction versus wall thickness, where contraction is here plotted towards the left. So on the x-axis, we have the Cauchy stress change due to contraction, which is more negative means more contraction. And as you can see, the more contraction to the left, the less the wall thickening. So it seems that the better the vessel is able to contract against the hypertensive stimulus, the lower the thickening is, and the better the homeostasis. And we can also compare this statistically, and if you do that using a multivariable regression model, the contraction comes out more strong than the inflammation. So summary up till now, what do we have? On the top, we have the C57 aorta, at the bottom 129 S6 aorta. And for the C57 aorta, we see a marked inflammatory response, but a very small contractile response to the angiotensin infusion. Whereas on the bottom, we see the opposite, basically, very strong contractile response and no inflammatory response, which we think leads to maladaptation, fibrosis, and little contraction on the top, and the opposite, so adaptation, no fibrosis, and a strong contraction on the bottom. So then we moved on and said, okay, can we further validate this contraction hypothesis, basically, or further dive deeper into it? And this was all despite the similar blood pressure I forgot to mention. So we then went on and continued by performing what we called wall stress analysis, which is essentially thick-walled modeling, where we can look at the wall stress distribution through the wall, RNA sequencing and gene ontology, and simulated growth and remodeling. So I'll now move on to the RNA sequencing part. So here you see just a small fraction of the genes that we looked at using the bulk RNA sequencing that we performed. As you can see, there's many of them, but I want to point your attention to, for example, IL-6, interleukin-6, which is a strong inflammatory marker. And the bars here show the fold change with respect to the baseline or normotensive value, with in black again the C57 mice and in gray the 129 mice. And you can see here a marked upregulation of IL-6 in the C57s and no change in the 129s. And the overbar denotes that there is actually a difference between those changes. So there's an interaction effect, so to say. So you can do this for many genes. I'm not going to go through all of them. One of them of interest here is MYH11, a contractile gene, which is indeed downregulated in the C57s and not changed in the C57 in the 129s. And the same holds for active-2 here on the right. And the collagens are all up, so which is consistent with the collagen production in both strains, but they are more up in the C57s and the 129s, which is consistent with the more exuberant collagen production there as opposed to the other strain. So this is all nice, but this is, as you can see, this is looking for a needle in a haystack, basically, because this is there is 30,000 genes quantified using our RNA sequencing. So ideally, we want to use something more sophisticated rather than looking at individual genes. And one of the ways to do this is to look at what's called a gene ontology analysis. So gene ontology analysis means that instead of looking at individual genes, we look at genes grouped by processes. I'm going to take a sip of water. So, and similar to the gene expression here, we can look at which processes are upregulated in one group versus another, or vice versa. So here on this plot and this bar chart, we see which processes are upregulated when we compare the hypertensive 129 strain mice versus the hypertensive C57 strain mice. And indeed, if we look at these processes, we see that contraction and muscle-related processes are upregulated in the 129 aorta as compared to the C57 aorta, which is consistent with our mechanical findings and our contraction findings. So contractility indeed seems to be the upregulated factor here. If we look in the different directions, so note the greater than or smaller than sign here, we see that in the C57 compared to the 129, there's an upregulation of immune responses and inflammatory responses, again suggesting that indeed perhaps there's an inflammatory response in the C57, but rather a contractile response in the 129s. And finally, what we can do is we can select processes of interest, such as muscle structure development and immune system processes, and we can actually see which genes that are part of these processes are up or down regulated, as are visualized on the left here in blue for an upregulation in C57 versus in red for an upregulation in 129 versus the other strain. So this basically confirmed our hypothesis, saying that, okay, in the C57 aorta, there seems to be an inflammatory response, but in the 129 aorta, there seems to be mainly a contractile response. So then we went on to what we called wall stress analyses, so thick wall bilayered analyses. So on a plot here, for example, you see for the normotensive groups, so without angiotensin infusion, the circumferential wall stress going from the lumen, so from the inside to the outside of the vessel. So this plateau here is the media, this plateau is the adventitia. And we see that the medial stress is higher than the adventitial stress, which is what you expect, because the adventitia is typically thought of as a shield for higher pressures. And we see in the 129 S6 aorta an adventitial stress of around 194 kilopascals. So if we then look at what happens after angio infusion in terms of passive mechanics, we actually see that neither of those mouse strains show an adventitial wall stress response that is homeostatic. So that is that reduces or that takes the wall stress back to baseline. So for the 129 aorta, we see in what you would call under adaptation. So the adventitial wall stress actually increases, as does the mean stress. But for the C57s, the adventitial wall stress decreases because of the exuberant thickening. There's just simply too much wall thickness to distribute the force. So the adventitial stress decreases to below the baseline level. However, this is passively. We know that these vessels can contract. So if we actually take into account the contraction of these vessels to Ang2 as measured, we actually see that the 129 S6 aorta, in that aorta, the adventitial stress was actually reduced back to the baseline level, whereas in C57, it is not, it is actually still below the baseline level. So this should suggest to us that adventitial homeostasis indeed may be a relevant factor in determining whether there's adventitial fibrosis going on. So just to illustrate how big these contractions actually are, these are the four groups of interest here. So on the top, we have the normotensive groups. On the bottom, we have the hypertensive groups. So the left column is the C57s. The right column is the 129s. And I want you to pay attention mainly to the bottom panel here, where you see the vessel contracting to angiotensin to angiotensin 2, with on the left the C57 and on the right the 126 aorta. And as you can see, there is a markedly stronger contractile response in the 129 aorta as compared to the C57 aorta. It's really more than a 50% on average inner diameter decrease, which is really remarkable. And of course, so the color here is the circumferential stress. So associated with this is also a marked circumferential stress reduction, as you would expect. So in the C57s, adventitial stress is not normalized after hypertension, whereas in the 129s, it is normalized. So finally, together with Marcus Latorre, we implemented the growth and remodeling model to look at the effect of contractility on fibrosis. So Marcus implemented a stress-dependent fibrosis or a remodeling term, as well as a contractility term. So what we did is we simulated a increase in systolic blood pressure together with the concomitant measured increase in contractility for the two groups, and kept all else equal, and then sought whether we could whether this difference merely in contractility could explain the difference between the groups. So this is the radius response that you get then, and these, more interestingly, are the medial thickness on the top and the adventitial thickness on the bottom. And as you can see, so the bar and whiskers are the measured data, and the line is the model response. We can indeed capture this adventitial, this marked adventitial thickening of the C57 aorta as compared to the 129 aorta, merely given a difference in their contraction, So if we could translate that to circumferential stress, indeed we see on the top here that in the 129 aorta, circumferential stress is almost maintained at the baseline level, whereas in the C57 aorta, you get a close over adaptation and the stress reduces to far below the baseline level. So this, well, we say predict here, but indeed this is in line with what we measured experimentally. So in the C57, based on the contractility, we expect a maladaptive remodeling, whereas in the 129 aorta, based on the contractility, we understand the adaptive remodeling. So this led us to conclude that contraction may actually facilitate aortic adaptation and prevent fibrosis or inflammation. And with that, I've come to the end of this talk. Of course, this was a lot of work, and there were a lot of people contributing to this. As you can see on the left here, so a big thank you to all of them. Of course, to Jay Humphrey for hosting me in his lab, and to all of the lab members for helping me. Also, a big thank you to my funders, the NWO, so the Dutch Organization for Scientific Research, and European Union. And thank you to all of you, for your attention, and I'm happy to take any questions.
Great talk, Bart. Thank you.
Thank you.
Are there any questions from the audience for the speaker?
I mean, maybe first of all, I want to congratulate you. That is a massive amount of work. I can't even believe it. This is one paper.
Well, in hindsight, it probably shouldn't have been, but yeah.
Yeah, it's funny, when I was in the lab, you could clearly sort of see, where people started out, which was like just biaxial tests, like sort of in the, I guess, late 90s, early 2000s. And then like every generation had to do new stuff. And while I was there, people were starting to do the active contraction. I was like, oh my God, where is this going to lead? But I guess you're the answer. Every generation of postdocs and students added yet another layer of analysis. But I mean, amazing work, beautiful. There's so many details to it that I have questions about. I'm going to let other people ask first. And I know that Matt prepared some questions. But I really love the holistic perspective that you took on this. We're trying to do this in the lab, but it requires a huge spectrum of skills and tools and experiences that's just really hard to find anywhere else. So anyways, congratulations. And also the visual presentation of your work is just fantastic.
Thank you.
You know, I can't help but congratulate you for that as well.
Thank you.
Okay, looks like we have a question. Trisha?
Yeah, hi. This is a really informative presentation. I just had a question. I think it was on slide 19.
Okay.
You were showing that there was a higher, thicker fiber deposition within the adventitia.
Yeah.
I was wondering, is there a specific like mechanical or biological reason behind that, where it was in that specific region, or am I missing something?
Good question. I don't know, to be fair. So I do know that we have observed as in different fibrosis models that the thicker fibers are typically seen in a fibrotic response. So that in that way it makes sense. But other than that, I have to owe you a full explanation to that. Yeah, again, as Matt said, this is just a gigantic bunch of data that we are starting to make sense of. Maybe Matt can comment.
I was actually going to add a comment. Yeah. So one thing, at least in the tissue engineering literature that's out there, when you see sort of remodeling of polymers into these so-called neo-tissues, there's been some observations where in cases of increased or heightened inflammation, that the collagen structure itself is a little bit different. And so the C57 in this case with a higher inflammatory, at least presence in that outer adventitial wall, that may be contributing somehow to the thickness of the collagen fibers. I would say that would be sort of an anecdotal comparison. I don't know, I mean, to the original question, a biological or biomechanical reason why it would be different, I'm not sure. But I would just sort of add that in as a potential comment.
Yeah, thanks.
Any other questions?
Don't be shy.
Okay, I'll ask a very general question. So how do you think that these differences between the different groups of mice you were looking at would translate onto human tissue? Like, do you think there's people out there that have a more inflammatory response and some of us have more of a contractile response? What is your
yeah, very good question. So I mean, obviously there is we also have different genetic backgrounds and hence indeed different responses. So yeah, I do think that there potentially are different responses to this, but on a more not to say a mouse strain level, but more a contractility level. I think this is really relevant also in the treatment of hypertension, because we typically treat hypertension, or actually many antihypertensive drugs reduce wall tension, relax the wall, and given these observations, that's maybe strange to do that, because yeah, because you may actually be impairing the homeostasis in the wall. But yeah, I mean, obviously there probably will be differential effects between people, and hence potentially even therefore different optimal treatments of people with different backgrounds because of this. Yeah. So as you may have gleaned from the introduction of this talk, this started out as a validation of a different study, where we were actually looking at the combined effect of hypertension and aging, and then stumbled upon differences between genotypes, and then actually, when we started looking at what do these differences entail other than a fibrotic versus non-fibrotic response, then we came across, okay, there's a marked contractile difference, and also that comes back if you look at the gene expression pattern and all of the other metrics. So I guess if you would design the study from scratch, you would probably, there's of course other ways to have a gradient in different levels of contraction between mice. For example, you can have knockouts of contractile proteins or partial knockouts, etc. So that would be the obvious way to prove a contractility difference. But it's just that, again, the path is not straight, but it's rather nonlinear, and that's why basically the methodology that I present is slightly unusual to prove an influence of contractility, if you're honest.
Thank you.
No worries, thanks.
Adrian is raising his hand.
Very good question. Are you still planning on doing the aging plus hypertension, or is that out of the picture?
We did actually. We published that first. So yeah, we did continue with the study and with, of course, the internal controls from that study. So so on a mixed background. So obviously the hypertensive response, as I already showed, is less than what we expected from Matt's earlier work. But given that, we still can compare the aging to the hypertension response, and what we actually see is that the aging, the contribution of aging or our model of aging is actually bigger than that of hypertension in terms of structural stiffening of the wall, and that's actually consistent with what has been observed in humans as well, where also people have done clever studies. I forgot how exactly to disentangle the two. And for example, you see in I think Marfan syndrome or other elastopathies that if you start out with an affected vessel, with a well, not a knockout, but a diseased vessel, that aging, that hypertension, sorry, has a smaller effect than if you start out with a healthy vessel. So that summarizes the study, but the study is out there in Journal of Hypertension if you wanna look it up.
Cool, thank you.
Manuel, you mentioned you had a comment or two, a question, maybe?
Yeah.
If there's not any from the audience, anyone else?
It's obviously not my area of expertise, but the one thing that I can't help but wonder about as a PI, as you did all this, you had a lot of help from people that were experts in various areas of this, right?
Yeah.
How are you gonna manage to do this kind of work moving forward? And I'm looking forward to being educated by you, because I really struggle, and luckily I obviously have colleagues that are on the call here, such as Matt, who have a pretty orthogonal expertise to mine. But I'm just curious, are you lining up a pretty broad spectrum of collaborators? Are you trying to build your own lab and do these things by yourself? I'm just curious, because now that you, I guess, tasted the blood, I presume you're going to do this moving forward, right? Like, do you not say that in English? That's maybe a German expression, but you know what I mean.
I know what you mean, yeah. Yeah, very good question. So I guess the answer is both. So indeed, on the one hand, have a diverse team of PhD students, so not only engineers, but also we have a medical student and also someone who studied natural sciences. So really make sure to have a variety of backgrounds there, but still then necessarily I mean, those people are young and need to be trained, so you still will need expertise. So I mean, so that's one of the reasons why I wanted to come back to Maastricht, is because we are actually a very small engineering department, and we're not in in an engineering faculty here. This is mainly a medical, we have a very big medical faculty that we're health sciences faculty that we're part of. So there's actually quite a lot of expertise on the non-engineering stuff around here in our cardiovascular research school. So indeed, for example, gene expression, I will collaborate with other people to do that. I mean, the engineering is mostly ourselves. If it's going to be more, if the engineering is at its limits here, we are close to Eindhoven University of Technology, which is an actual technical university which has a lot more engineering. But yeah, I strongly believe in working together with people within in the institute and abroad, or well, abroad meaning in the same country, but also not in the same country. So yeah, more and more, I mean, if you want to do a study like this, you cannot, as a starting PI, just not do it on your own, and you shouldn't try. You should just embrace all the collaborations that there are and cherish them, and I think that's the way to do it.
Thank you, appreciate it.
So don't worry about not having all the expertise. I also don't.
Yeah, I think that's a really, you phrased that very well, Bart. I think the collaboration is certainly important. And to use the word cherish, I think that adds a nice sense to the word collaboration. So, I had a quick question, sort of about the modeling efforts, and if you had thought about, how to potentially bridge the gap between some of the growth and remodeling or the simulations and the transcriptomics data from the RNA sequencing. With all of these, flourishing techniques in terms of gene expression analysis and transcriptomics, do you think that there's a space to combine these two? And if so, have you thought about this, or just kind of your general sense on it?
So yes, I thought about it, and this comes back to, okay, we need collaborations. So for example, I know that at Jay's lab, Linda Irons and Anna Estrada are working on these kinds of modeling approaches. So yeah, certainly there is room for this, and this is really relevant to do. So yes, but yeah, for now it's not going to be my main focus, just because I have to stop somewhere basically, and a day only has 24 hours. But yeah, certainly there is room for that, but again, I would need to reach out to people to do that. Yeah.
Looks like there's a question in the chat from Saurav. It says, how did you quantify the histology images to get the mass fractions of the constituents?
Good question. So at the Humphrey lab, we used a code. I don't know, probably Matt, did you write it? Or
most of it?
Most of it, yes. So a code that basically looks at the colors in the images. So briefly, it converts the images to the hue, saturation, lightness space rather than RGB. You can determine the colors of the various constituents, and then using some smart thresholds and subtractions. After, of course, standardizing all of the images that you've taken, so to ensure there's no experimental variability there, that will give you an estimate of the mass fractions from the histology images. But it's I mean, I would really recommend always have your own controls. Ideally, also stain all of the images in one batch, because differences between batches of histology staining will also potentially bias your results. But that's in a nutshell how we did that. So colorimetrically. That's the very short answer.
Great. I hope that answered. Okay, great. Yeah, that answered his question. Anything else? Any comments from anyone? Questions?
There's one question in the chat, maybe. Oh no, I just commented. I have a question moving forward. What are you going to be working on? Similar problems, different problems?
Similar, yeah. So we are working on developing a setup to look at the physical elasticity of vessels. So not do quasi-static loading, but rather very quick loading, and see how that influences material behavior, and see whether we can capture that using viscoelastic constitutive models, and also do that during contraction and relaxation, etc. And as you can imagine, that in itself is quite challenging to capture that all into one proper descriptive model is not easy, but very interesting, because it explains many phenomena, potentially also explains differences that are observed between in vitro and in vivo experiments, because typically what we call in vitro is very slowly inflated, whereas in vivo is of course very quickly. So just to give you an idea, a mouse heart beats 10 times a second. So you if you want to have a physiological loading rate in vitro, you want to have a base frequency of 10 hertz with the harmonics on top of that. So that's quite high. So it's quite fast tracking, and there's all kinds of issues going on in getting that straight in an experimental setup. But very nice work. Koen van der Laan, who I think is on the call, who was on the call earlier, he is doing a lot of work on getting that setup up and running here. And then one of the other things that I'm really interested in is human data, and seeing whether we can, to some extent, which is by definition more limited, do these things in humans. See whether we can fit constitutive models to human pressure and diameter measurements, and see whether we can say anything about pathologies using those measurements, etc. So it's shifting a little bit more towards so on the one hand physical elasticity, and on the other hand human measurements. But it's
exciting stuff.
My problem is that I find everything interesting, so that's the biggest pitfall of the science.
Probably strength and weakness, yeah.
Yeah.
Okay. Last minute comments, questions. We have just here in the final few minutes. Anybody?
Nothing, again, just really, I forgot to say it before, but second Manuel's comment that it's such a complete study, so many moving parts. I think that's very motivating for myself, for example, trying to do things that really touch on these different points.
Yeah, thanks. That's great to hear.
Yeah, absolutely. Let me stop the recording and then.
Yeah, so